Okay, next double membraned organelle that we're going to deal with is called a mitochondrion. This is one of the words that is most commonly misspelt. So mitochondrion is the singular. So if you're talking about one, you're talking about a mitochondrion. You probably won't get penalised too much. The term that everybody knows is mitochondria, which is usually talking about more than one. Now overall, mitochondrions' main role is to release. Now that's really important. It's not making energy, it's releasing energy by, and again, really important, it's aerobic respiration only, which is by far the more efficient sort of respiration. So you'll very often see these in textbooks referred to as the powerhouse of the cell. They're releasing energy through aerobic respiration. And the form in which they're releasing it in is ATP. We'll talk about that again in a minute. So these are sort of small lozenge-shaped or sausage-shaped inclusions, little organelles that have a particular role. Not really visible down a microscope, except as they just look like little specks, little lines inside the cytoplasm. Again, we have to go to an electron micrograph to see them properly, and I'm hoping this is quite visible. So again, we can see that we have two membranes, one there, one there. And if we follow this membrane round, we can see that it's folded right into the middle of the organelle and then comes back out. So the outer one is nice and smooth, but the inner one is folded. And just the way that the section of this mitochondrion has been sliced, obviously you can't see where you're slicing a mitochondrion, so they never look terribly tidy, electron micrographs. Some of them you can see sort of bits of membrane that don't look like they're connected to the outside, so they almost look as though they've got lines across them. So outer membrane nice and smooth, inner membrane folded, and folding generally means an increased surface area for something to happen, and what's happening in this is aerobic respiration, so it's an increased surface area for the enzymes involved in that. And then in between these folds we've got this sort of liquid full of enzymes called the matrix. So again, I'm just going to draw a little outline, put all the bits on that you need to know, and deal with the functions of each part. So, mitochondrion, start off by drawing a nice smooth sausage shape, and we're going to put the folds on the inner membrane. So we've got an outer membrane and an inner membrane, and it's not just a wavy line, they are actual real folds, which is why I'm doing them quite sort of sharp and spiky. So we could see both membranes on that electron micrograph. With those folds. And they're almost like finger-like projections into the middle. So we've got the outer membrane, the inner membrane, and these folds are called cristae. Now again, one would be a crista. I've got the label to 2, so I'm going to call them cristae with an E on the end. And these are folds that increase surface area. So, what are they increasing the surface area of? Well, actually, inside of this membrane are what we call stalked particles. I'm just going to draw them as lollipop sticks. And in upper sixth, you'll be doing what these little lollipop sticks do in a huge amount of detail. So these are an enzyme embedded into the membrane. So the more surface area you've got, the more enzymes you've got. So these are what we call stalked particles. They're actually the enzyme that manufactures ATP. Enzyme. And that enzyme is called ATP, because that's what it makes. Synthetase. And again, synthetase, you talk about synthesis, so this is making ATP. So this generates ATP, which is an energy-rich molecule. So this energy is going to be our ATP. And when we do membranes, we'll do sort of, how proteins are embedded in them, and ATP synthetase is a really good example of a membrane that's got an enzyme embedded in it. We also said that this gap in the middle is called the matrix. So this is a fluid containing enzymes. And again, when we do respiration, all the reactions in upper sixth, you'll find out what enzymes are actually in there. It's enough to know that there are enzymes in there for the minute. Mitochondria have another couple of unusual features in that they also have a circular loop of DNA. Now this DNA is just like the DNA of a prokaryote. It's kind of floating loose in this matrix. And so this has led to certain scientists proposing an endosymbiotic theory. That means endosymbiotic means that both things benefit. So this is thought to have been an early prokaryote that got engulfed by a larger cell. That larger cell thought, oh great, this prokaryote that I've just engulfed is releasing energy, I can use that. And of course the prokaryote gains a bit of protection from that larger cell. And has retained this circular DNA, which is just like that of bacteria. And it also allows mitochondria to self-replicate. So it doesn't actually need the cell's instructions from its nucleus to make more copies of itself. It can just happily replicate itself. And in addition, in the matrix, it also has ribosomes. And these are small ribosomes, so they're smaller than the ones that are sort of outside in the cytoplasm. And these are used for protein synthesis, with again the synthesis referring to making proteins. So mitochondrion, some bacterial sort of features, these small ribosomes, the circular DNA. You need to know that it's got a matrix, outer and inner membrane, folds on the inner membrane that increase the surface area, and these stalked particles that generate ATP, and of course those are found all over the place, and the ribosomes are found all over the place. Overall, releasing energy by aerobic respiration. So cells that do a lot of metabolic reactions that are highly energy requiring, things like muscles and liver, often have a lot of these mitochondria. Okay, that's it for mitochondria.